Good evening. In this session, we'll be dealing with Introduction to Embryology. So, what is embryology? It deals with prenatal stages of development from fertilization of ovum to the birth. So, we can study it in three different formats. Descriptive embryology, where we are going to study the complete development of one species, example humans, what we are going to study in first year MBBS. Then comparative embryology, where we are going to compare the developmental stages of different species, and experimental embryology, where we are going to investigate the sequence of developmental events by experiments and chemical induction. Then there is a term called ontogeny, which is like complete life cycle of an organism, like prenatal development, birth, postnatal development, growth, maturity, then after that a stage of regression, then senility and death. So it's a complete life cycle of an organism is called ontogeny. So if we say it is a gradual process of expression of blueprint of life in genes. Then phylogeny. So this phylogeny deals with evolutionary history and relationship among the organisms. So there is a term called ontogeny repeat phylogeny. We'll see the example, then we'll understand. If we see the development of human kidney, so initially there will be formation of pronephric kidney in the region of neck, so which is seen in some primitive fishes and cyclostomes. So later this disappears and there will be formation of mesonephric kidney in the lower part of the thoracic region and lumbar region. So this is in amphibians. And then ultimately there will be formation of a metanephric kidney in the lumbosacral region, which is retained as a permanent kidney in humans. So if you see this, the human body is undergoing stages of fishes, amphibians, and then reaching the mammals. So this is what it means, ontogeny repeats phylogeny. Now if you observe, the development is a process of conversion of a single cell structure to a complex multicellular organism. It involves two different steps. One is the growth, other one is differentiation. So if you see growth is mere increase in the number and size of the cells. So it is of three types. Multiplicative growth, where increase in cell number by cell division occurs, like we see in the epidermis of the skin and the intestinal epithelium. Then auxetic growth, it is increase in cell size, like we see in the ovum. Then if we see accretionary growth, increase in the intracellular substance, like we see in the cartilage and bone. Multiplicative growth, auxetic growth, and accretionary growth. Then if we come here, differentiation, it is a process of cell transformation to acquire specific character and function, like a specialized tissue. That is called differentiation. So initially the cells are totipotent. They produce entire embryo under favorable conditions, like initially after fertilization. If we see the first two cells which are formed after the cleavage, they are totipotent. They can completely produce an embryo. So then later on, after multiple divisions, they become pluripotent, where cells can produce specific tissues only. Okay. So then what happens? There will be a chemodifferentiation, which is induced by organizer. So organizer, if you take in the human embryos, we call the organizer as primitive streak, which is the primary organizer. So primitive streak induces the development of notochord and secondary mesoderm. So this notochord acts as the secondary organizers, which induces the formation of neural tube from the neuroectoderm. And this neural tube acts as a tertiary organizer, which will induce the formation of somates. So primary, secondary, and tertiary. So how these organizers will work? They act by two processes. One is evocation and individuation. Evocation is liberation of chemical substance called evocator, and this evocator will cause individuation. That is, through the interaction between the organizer and the host, there will be formation of different tissues. So after the chemodifferentiation comes the histodifferentiation. So at this stage of chemodifferentiation, there is no change in the structure actually. So there is only chemodifferentiation, that chemicals are being secreted and there are interactions. After this stage comes the histodifferentiation, where there is structural change in the cells, which will definitely look like some specified tissue. Then they will form organs, which results in organogenesis, and ultimately there will be functional differentiation. So totipotent, pluripotent, then chemodifferentiation by organizers through evocation and individuation, in histodifferentiation, then organogenesis and functional differentiation. So then if you see the periods of embryology, the germinal period from the fertilization till the first 3 weeks of development. So this is the germinal period. Then embryonic period from 4th to 8th weeks of development, and the fetal period from the 3rd month, that is from 9th week till the termination or completion of the pregnancy. Germinal period first 3 weeks, embryonic period 4th to 8th week, fetal period from 9th week to termination of pregnancy, or 4th month to 3rd month to termination of pregnancy. So here if you see the germinal period 0 to 2nd and 3rd weeks, so up to 3rd week, and then 4th to 8th week will be embryonic. From 9th week onwards it will be the fetal period. So if we see different things, prenatal and postnatal. In the prenatal, so from fertilization till the 3rd week, so after the completion of 3rd week, 4th to 8th week will be the embryonic period. The fetal period is from the 9th week till the birth. So till then we call it as fetus. So after that it is newborn. And if you see postnatal period, neonate is up to first 4 weeks after the birth. Infant is 1 month to 1 year. Child is more than 1 year to 12 years. Puberty is between 12 years to 16 years. Adolescent is 16 to 20 years. Adult is 20 to 40 years. Middle age is 40 to 60 years. And old age is beyond 60 years. So these are different periods. So few authors say it is first 2 weeks, few authors say it is first 3 weeks, but standard is first 3 weeks will be the germinal period, then from 4th to 8th week will be the embryonic period, and then from 9th week onwards till the termination of pregnancy, that is the birth, will be the fetal period. So here we call it zygote, here we call embryo, then we call it as fetus. So now coming to the cell cycle, just to have refreshing our knowledge with the cell cycle. So there is G1 phase, S phase, G2 phase, and M phase. So M phase is the mitotic phase where cell division is occurring. So G1 phase, what are the main things happening in G1 phase is cytoplasm increases in volume, damaged DNA gets repaired. In the S phase, there is replication of the DNA. In the G2 phase, it is like checkpoint for the conformation of duplicated chromatin. And ultimately mitotic phase, you can see here the mitotic phase is having prophase, metaphase, anaphase, and telophase, in which there is successive division. So this is the cell cycle: G1 phase, S phase, G2 phase, and the M phase. M phase is having this mitotic phase in which there is prophase, metaphase, anaphase, and telophase. So there are two types of divisions like mitosis, equatorial or equational division, and then meiosis, which is the reductional division. Let us compare and contrast. We are not going into see the details of all these cell cycle. We'll just try to highlight the compare and contrast between the mitosis and meiosis. You can see here all cells of the body undergo mitosis. Meiosis is seen only in germ cells. It is equational division. Mitosis is an equational division, whereas meiosis is reductional division. So if you see in the prophase, there is no crossover of genetic material, no synapses. But if you see in the meiosis, there is crossover of genetic material and synapses occurs. In metaphase, there is no chiasmata formation. Chromosomes arrange at equator. So chiasmata formation is present. Homologous chromosomes arrange on either side of the equator. So in anaphase, centromere divides. Chromatids move to opposite pole. Here there is no division of centromere. Whole chromosome moves to the opposite pole. So telophase, daughter cells with same number of chromosomes, 46. Here daughter cells with haploid number of chromosomes. There is reduction in the chromosomal number to half. So number of daughter cells formed in mitosis will be 2. Number of daughter cells formed in meiosis will be 4. So now, when we apply this embryology to understand clinical importance, there is a first major step which is going wrong. So what is this disjunction? It is a normal process, usual separation of chromosomes in first meiotic division or sister chromatids in second meiotic division. This is normal. But when we go to non-disjunction, abnormal segregation results in cells receiving less number of chromosomes or extra chromosome. So in which fetus with an abnormal number of chromosomes, they'll have trisomies or monosomies. Example, Down syndrome, trisomy of 21 chromosome. Trisomy of 21st chromosome results in Down syndrome. Klinefelter syndrome, extra X chromosomes in males. Okay, 47 XXY. Klinefelter. Turner syndrome, lack of Y chromosome in phenotypic female, 45X. Okay, so these are few syndromes which result from non-disjunction. So that is why we study the embryology. So whatever goes wrong in the development is due to some kind of chromosomal abnormalities or genetic abnormalities. Now if you observe, 3 to 4% of the live births, they suffer from birth defects like cleft lip, ventricular septal defect, atrial septal defect, cleft palate. So to understand the gross anatomy of a body, that means how the adult structure of the heart, adult structure of the lung, adult structure of the face, adult structure of the eye has been formed, and histological structures of the tissues, we need to understand the embryology, from which cell lineage they are being developed. Then mutation of genes controlling the development. So if any gene which is controlling the normal process is mutated, then it results in disease. So that is why we need to understand the basic genes which are helping in the normal development. Any change in those genes, like mutations, will result in a disease. Then reproductive medicine, most evolving for better practice for the well-being of mother and newborn. So we need to understand the complete developmental process, because we have ultrasound which will help us to see the developing baby. So at any given point of time, if we find any kind of developmental defects which will not sustain with life, then we need to advise for medical termination of pregnancy. And for infertility cases also, we need to identify the cause. Then we can go for advanced techniques like in vitro fertilization or intrauterine insemination. Then again, in prenatal diagnosis, and to have certain therapy for fetus. So we have certain tests like amniocentesis, chorionics, villus sampling at different stages of development of the fetus, to so that we can diagnose this earlier syndromes which I told: Down syndrome, Klinefelter's, Turner's, Edwards, Pottow's. All these syndromes can be diagnosed. So this concludes introduction to embryology. From the next class onwards, we'll deal with the gametogenesis, ovarian cycle, menstrual cycle, fertilization, and then stage-wise development: first week, second week, third week, like that. Okay. Thank you.